सो हेलो गाइस, कैसे आप सब लोग। माई सेल्फ सिद्धिकांत सिक्वाल और आज मैं स्वागत करता हूँ आप सभी का हमारा अपना पहला सा चैनल एसक्यूएल जे पे इन द सीरीज ऑफ एनसीईआरटी लीडिंग लाइन बाय लाइन। तो आज हम शुरू करने वाले हैं एक्सलिटरी प्रोडक्ट्स एंड देयर एलिमिनेशन। तो चलिएगा शुरू करते हैं। एनिमल accumulate ammonia, urea and uric acid, carbon dioxide, water and ions like Na plus, K plus, Cl minus, phosphate, sulfate, etc. either by metabolic activities or by other means like excess injection. These substances have to be removed totally or partially. In this chapter, you will learn the mechanism of elimination of these substances with a special emphasis on a common nitrogenous waste. Ammonia, urea, and uric acid are the major forms of a nitrogenous waste excreted by the animals. Ammonia is the most toxic form of a form and requires a large amount of water for its elimination, whereas uric acid, being the least toxic, can be removed with the minimum loss of water. The process of excreting ammonia is ammonotelism. Many bony fishes, aquatic amphibians, and aquatic insects are ammonotelic in nature. Ammonia, as it is readily soluble, is generally excreted by a diffusion across body surface or through gill surface in fish as ammonium ions. Kidney do not play any significant role in this removal. In its removal, terrestrial adaptation necessitated in the production of a lesser toxic nitrogenous waste like urea and uric acid for conservation of water. Mammals, many terrestrial amphibians and marine fishes mainly excrete urea and are called ureotelic animals. Ammonia produced by metabolism is converted into urea in the liver of these animals and released into the blood, which is filtered and excreted out by the kidneys. Some amount of urea may be retained in the kidney matrix of the some of these animals to maintain a desired osmolality. Reptiles, birds, land snails, and insects excrete nitrogenous waste as a uric acid in the form of pellet or paste with the minimum loss of water and are called ureotelic animals. A survey of animal kingdom present a variety of skeletal structures in most of the invertebrates. These structures are simple tubular forms, whereas vertebrates have a complex tubular organs called kidney. Some of these structures are mentioned here. For Photonephridia or frame cells are skeletal structures in a platyhelminths, that is flatworms, example Planaria. Lotif Lotifers, some annelids, and the cephalochordates, that is Amphioxus. Photonephridia are primarily concerned with ionic and fluid volume regulation, that is osmoregulation. Nephridia are a tubular excretory structures of earthworms and other annelids. Nephridia help to remove the nitrogenous waste and maintain a fluid and ionic balance. Malpighian tubules are the excretory structures of most of the insects, including cockroaches. Malpighian tubules help in the removal of nitrogenous waste and osmoregulation. Antennal glands or green glands perform a excretory function in the crustacean-like ponds. 16.1 Human excretory system. In human, the excretory system consists of a pair of a kidney, one pair of ureter, a urinary bladder, and a urethra. Kidney are reddish brown bean-shaped structures situated between the level of a large thoracic and third lumbar vertebra close to the dorsal inner wall of the abdominal cavity. Each kidney of the abdominal each kidney of an adult human measures 10 to 12 cm in length, 5 to 7 cm in width, and 2 to 3 cm in thickness, with an average weight 120 to 170 gram. Towards the center of the inner concave surface of the kidney is a notch called hilum, through which ureteral blood vessels and nerves enter. Inner to the hilum is a broad funnel-shaped space called the linear pelvis with a projections called calyxes. The outer layer of the kidney is tough capsule. Inside the kidney, there are two zones, an outer cortex and inner medulla. The medulla is divided into few conical masses, that is medullary pyramids projecting into the calyxes, singular calyx. The cortex extends between the medullary pyramid as a linear columns called columns of Baltini. Each kidney has a nearly 1 million complex tubular structures called nephrons, which are the functional unit. Each nephron has two parts, the glomerulus and the linear tubule. Glomerulus is a tuft of capillaries formed by the afferent arteriole, a fine branch of a linear artery. Blood from the glomerulus is carried away by an efferent arteriole. The linear tubule begins with the double-walled cup-like structures called Bowman's capsule, which enclose the glomerulus. Glomerulus, along with Bowman's capsule, is called as Malpighian body or a linear corpuscle. The tubular the, tu- the tubule continues further to form a highly coiled network that is proximal convoluted tubule. PCT. A heparin-shaped Henle's loop is the next part of the tubule which has descending part and ascending limb. The ascending limb continues as another highly coiled tubular lesion called distal convoluted tubule DCT. The DCT of a many nephrons opens into a straight tube called collecting duct, many of which converge and open into linear pelvis through medullary pyramid in the calyces. The Malpighian corpuscle (PCT) and the DCT of the nephrons are situated in the cortical region of the kidney, whereas the loop of Henle dips into the medulla. In majority of nephrons, the loop of Henle is too short and extends only very little into the medulla. Such nephrons are called cortical nephrons. In some of the nephrons, the loop of Henle is very long and runs deep into the medulla. These nephrons are called juxtamedullary nephrons. The efferent arteriole emerging from the glomerulus forms a fine capillary network around the linear tubule called a pelti called a peritubular capillaries. A main vessels a main vessel of this network runs parallel to the Henle's loop, forming a U-shaped vasa lecta. Vasa lecta is absent or highly reduced in a cortical nephrons. 16.2 Urine formation. Urine formation involves three main processes, namely glomerular filtration, reabsorption, and secretion, that takes place in a different part of the nephron. The first step in a urine formation is filtration of blood, which is carried out by a glomerulus and called as glomerular filtration. On an average, 1100 to 1200 ml of blood is filtered by the kidney per minute, which constitutes roughly one-fifth of the blood pumped out by each ventricle of the heart in the minute. A glomerular capillary blood pressure causes the filtration of a blood through three layers, that is, the endothelium of a glomerular blood vessel, the epithelium of Bowman's capsule, and basement membrane between these two layers. The epithelial cells of a Bowman's capsule called podocytes are arranged in an intricate manner so as to leave some minor spaces called filtration slits or a slit pores. Blood is filtered so finely through these membranes that almost all the constituents of the plasma except the protein pass on into the lumen of the Bowman's capsule. Therefore, it is considered as the process of ultrafiltration. The Bowman's the amount of the filtrate formed by the kidney per minute is called glomerular filtration rate GFR. GFR in a healthy individual is approximately 125 ml per minute, that is 180 liters per day. The kidney have a built-in mechanism for the regulation of glomerular filtration rate. One such efficient mechanism is carried out by the juxtaglomerular apparatus, that is JGA. JGA is a special sensitive lesion formed by the cellular modifications in the DCT and afferent arteriole at the location of their contact. A fall in a, a fall in a GFR can activate the JG cells to release renin, which can stimulate the glomerular blood flow and thereby the GFR back to normal. A comparison of the a comparison of the volume of the filtrate formed per day, 180 liters per day, with that of the urine released, 1.5 liters, suggests that nearly 99% of the filtrate has to be reabsorbed by the renal tubules. This process is called reabsorption. The tubular epithelial cells in a different segments of a nephron perform this either by an active or passive mechanism. For example, substances like glucose, amino acids, Na+, etc. in the filtrate are reabsorbed actively, whereas the nitrogenous waste are absorbed by the passive transport. Reabsorption of water also occurs passively in the initial segments of the nephron. During urine formation, the tubular cells secrete substances like H+, K+, and ammonia into the filtrate. Tubular secretion is also an important step in the urine formation as it helps in the maintenance of the ionic and acid base balance of the body fluid. 16.3 Functions of tubules. Proximal convoluted tubule PCT. That is, PCT is lined by a simple cuboidal plus bordered epithelium which increases the surface area for reabsorption. Nearly all the essential nutrients and 70 to 80% of the electrolytes and water are reabsorbed by this segment. PCT also helps to maintain the pH and ionic balance of the body fluids by the selective secretion of hydrogen ions and ammonia into the filtrate and by absorption of a HCO3- from it. Then Henle's loop. That is, reabsorption is minimum in its ascending limb. However, this lesion plays a significant role in the maintenance of a high osmolality of a medullary interstitial fluid. The descending limb of a loop of Henle is permeable to water but almost impermeable to electrolytes. This concentrates the filtrate as it moves down. The ascending limb is impermeable to water but allows the transport of electrolyte actively or passively. Therefore, at its contracted filtrate pass upward, it gets diluted due to the passage of electrolyte to the medullary fluid. Distal convoluted tubule (DCT). That is, conditional reabsorption of Na plus and water takes place in this segment. DCT is also capable of reabsorption of HCO3- and selective secretions of hydrogen and potassium ions and NH3 to maintain the pH and sodium potassium balance in blood. Then collecting duct. These long ducts extend from the cortex of the kidney to the inner part of the medulla. Large amount of water could be reabsorbed by this lesion to produce the concentrated urine. This segment allows passage of a small amounts of urea into the medullary interstitium to keep up osmolality. It also plays a role in the maintenance of pH and ionic balance of blood by the selective secretion of H plus and K plus ions. 16.4 Mechanism of concentration of the filtrate. Mammals have the ability to produce a concentrated urine. The Henle's loop and the Vasaretta play a significant role in this. The flow of filtrate in the two limbs of Henle's loop is in opposite direction and thus forms a counter current. The flow of blood through the two limbs of Vasaretta is also in a counter current pattern. The proximity between the Henle's loop and the vasa recta as well as the counter current in them help in the maintaining and increasing osmolality towards the inner medullary interstitium, that is from 300 osmol per liter in the cortex to about 1200 more osmol per liter in the inner medulla. This gradient is mainly caused by the NaCl and urea. NaCl is transported by the ascending limb of Henle's loop, which is exchanged with the descending limb of a vasa lecta. NaCl is returned to the interstitium by the ascending portion of the vasa lecta. Similarly, small amounts of urea enter the thin segment of the ascending limb of a Henle's loop, which is transported back to the interstitium by the collecting tubule. The above descending transport of the substance fasciculates by the special arrangement of Henle's loop, and vasa lecta is also called the countercurrent mechanism. This mechanism helps to maintain the concentration gradient in the medullary interstitium. Presence of such interstitial gradient help in an easy passage to water from the collecting tubule, thereby concentrating the filtrate, that is urine. Human kidney can produce a urine nearly 4 times concentrated than the initial filtrate formed. 16.5 Regulation of kidney function. The functioning of the kidney is efficiently monitored and regulated by the hormonal feedback mechanism involving the hypothalamus, JGA, and to the certain extent the heart. Osmoreceptors Osmoreceptors in the body are activated by the changes in blood volume, body fluid volume, and ionic concentration. An excessive loss of a fluid from the body can activate these receptors, which stimulate the hypothalamus to release the antidiuretic hormone, that is ADH, or vasopressin from the neurohypophysis. ADH facilitates the water reabsorption from a lateral part of the tubule, thereby preventing the diuresis. An increase in the body fluid volume can switch off the osmoreceptors and suppress the ADH release to complete the feedback. ADH can also affect the kidney function by its consist, cons, constrictory effect on blood vessels. This causes an increase in the blood pressure. An increase in the blood pressure can increase the glomerular blood flow and thereby the GFR. The JGA plays a complex regulatory role. A fall in the glomerular blood flow or a glomerular blood pressure or a GFR can activate the JG cells to release renin, which converts the angiotensin in blood to angiotensin first and the further to angiotensin second. Angiotensin second, being a powerful vasoconstrictor, increases the glomerular blood pressure and thereby GFR. The angiotensin second also activates the adrenal cortex to release the aldosterone. Aldosterone causes the reabsorption of Na plus and water from the distal part of the tubule. This also leads to an increase in the blood pressure and GFR. This complex mechanism is generally known as the renin-angiotensin mechanism. An increase in the blood flow to the atria of the heart can cause the release of atrial natriuretic factor, that is ANF. ANFs can cause a vasodilation, that is dilation of a blood vessels, and thereby decrease the blood pressure. ANF mechanism therefore acts as a check on the renin angiotensin mechanism. 16.6 Mictulation. Urine formed by the nephrons is ultimately carried to the urinary bladder, where it is stored till the voluntary signals is given by the central nervous system CNS. This signals is initiated by the stretching the urinary bladder as it gets filled with the urine. It responds the stretch receptors on the walls of the bladder send signals to CNS. The CNS passes on the motor messengers to initiate the concentration of smooth muscles of the bladder and stimulates the relaxation of the urethral sphincters, causing the release of the urine. The process of release of urine is called mictulation, and the neural mechanism causing it is called mictulation reflex. An adult human excretes on an average 1 to 1.5 liters of urine per day. A urine formed is a light yellow colored watery fluid which is slightly acidic, that is pH 6, and has a characteristic odor. On an average, 25 to 30 gram of urea is excreted out per day. Various conditions can affect the characteristics of a urine. Analysis of urine helps in the clinical diagnosis of many metabolic disorders as well as malfunctioning of the kidney. For example, presence of glucose gas is glycosuria and ketone bodies, that is ketonuria in a urine are indicative of diabetes mellitus. 16.17 Role of other organs in excretion. Other than kidney, lungs, liver, and skin also helps in the elimination of excretory wastes. Our lungs remove large amount of CO2, that is approximately 200 ml per minute, and also significant quantities of water every day. Liver, the largest gland in our body, secretes the bile containing substances like bilirubin, biliverdin, cholesterol, degrades the steroid hormones, vitamins, and drugs. Most of these substances ultimately pass out along with the digestive waste. The sweat and the sebaceous gland in the skin can eliminate certain substances through their secretion. Sweat produced by the sweat glands is a watery fluid containing NaCl, small amounts of urea, lactic acid, etc. Though the primary function of the sweat is to facilitate a cooling effect on the body surface, it is also helps in the removal of some of the waste mentioned above. Sebaceous gland eliminate certain substances like sterols, hydrocarbons, and waxes through the sebum. This secretion provides a protective oily covering for the skin. Do you know the small amount of nitrogenous waste could be eliminated through saliva too? 16.8 Disorders of excretory system. Malfunctioning of a kidney can lead to the accumulation of a urea in blood, a condition called ulemia, which is highly harmful and may lead to the kidney failure. In such patients, urea can be removed by a process called as hemodialysis. During the process of hemodialysis, the blood drained from the convenient artery is pumped into the dialyzing unit called artificial kidney. Blood drained from the convenient artery is pumped into the dialyzing unit after adding an anticoagulant like heparin. The unit contains a coiled cellophane tube surrounded by a fluid that is dialysing fluid having the same composition as that of a plasma except the nitrogenous waste. The, the polar cellophane, cellophane membranes of the tube allows the passage of molecules based on the concentration gradient. As a nitrogenous waste are absent in the dialysing fluid, these substances near freely move out, thereby clearing the blood. The cleared blood is pumped back to the body through a vein after adding antiheparin to it. This method is a boon for thousands of ulemic patients all over the world. Kidney transplantation, kidney transplantation is the ultimate method in the correction of the acute renal failure, that is kidney failure. A functioning of a kidney is used in the transplantation from the donor, preferably to close relative to minimize its chances of a rejection by the immune system of the host. Modern clinical procedures have increased the success rate of the of such a complicated techniques. Linear calculi. Stone or a insoluble mass of a crystallized salt that is oxalates, etc. forms within a kidney. Glomerulo glomerulonephritis, that is inflammation of a glomeruli in kidney. Inflammation of glomerular of kidney. Okay, summary. Many nitrogen containing substances, iron, CO2, water, etc., that accumulate in the body have to be eliminated. The nature of a nitrogenous waste formed and their excretion vary among animals, mainly depending on the habitat, that is, availability of a water. Ammonia, urea, and uric acid are the major nitrogenous waste excreted. Photonephridia, nephridia, Malpighian tubules, green glands, and the kidney are the common excretory organs in animals. They not only eliminate nitrogenous waste but also helps in the maintenance of ionic and acid-base balance of the body fluids. In humans, excretory system consists of one pair of kidney, a pair of ureter, a urinary bladder, and a urethra. Each kidney has over a millions of tubular structures called nephrons. Nephrons is the functional unit of kidney has a two portions, glomerulus and a linear tubule. Glomerulus is a tuft of capillaries formed from a afferent arteriole, fine branches of a linear artery. The linear tubule starts with a double-walled double double-walled woman's capsule and is further differentiated into a proximal convoluted tubule, that is PCT, Henle's loop HL, and a distal convoluted tubule DCT. The DCT of a many nephrons join to a common collecting duct, many of which ultimately opens into the linear pelvis through the medullary pyramid. The woman's capsule encloses the glomerulus to form a Malpighian or a linear corpuscles. Urine formation involves three main processes, that is filtration, reabsorption, and secretion. Filtration is a non-selective process performed by a glomerulus using a glomerular capillary blood pressure. About 1200 ml of blood is filtered by a glomerular per minute to form a 125 ml of filtrate in the woman's capsule per minute, that is GFR. JGA, specialized portion of the nephrons, play a significant role in the regulation of GFR. Nearly 99% of early absorption of the filtrate takes place through the different parts of the nephron. PCD is the major site of reabsorption and the selective secretions, that is HL, primarily helps to maintain osmolar gradient, that is 300 mole osmol per liter to 1200 mole osmol per liter within the kidney interstitium. DCT is the collecting duct allow the extensive absorption of water and certain electrolyte, which helps in the osmoregulation, that is H plus, K plus, and NH3 could be secreted into the filtrate by the tubules to maintain the ionic balance and pH of body fluid. A countercurrent mechanism operates between the two limbs of a loop of Henle's and those of vasa recta capillary parallel to Henle's loop. The filtrate gets concentrated as it moves down to descending limb but is diluted by the ascending limb. Electrolytes and urea are retained in the interstitium by this arrangement. DCT and a collecting duct concentrate the filtrate about 4 times, that is from 300 mole osmol per liter to 1200 mole osmol per liter. An excellent mechanism of conservation of water. Urine is stored in the urinary bladder till a voluntary signals from CNS carries out its release through the urethra, that is methylation. Skin, lungs, and liver also assist in excretion. Here we complete the excretory products and their elimination chapter. सो आपको ये चैप्टर कैसा लगा कमेंट्स में ज़रूर बताना, और ये आपको कहीं भी ट्रैवल और करते हो आप तो फटाक से एनसीईआरटी आपको रिवाइज हो जाएगी. सो थैंक यू फॉर वॉचिंग दिस वीडियो एंड मीट यू सून.